GMOs. I'm sure you've heard about them in the news, in debates about our crops, and as a warning from your mother at the grocery store. With so many conflicting ideas about GMOs, I bet you have one question. What are they?
When most people think of GMOs, this comes to mind. Food. But what if I were to tell you there's a lot more to GMOs than just this?
GMO is short for genetically modified organism, and is by definition an organism whose genome has been engineered in the laboratory in order to favor the expression of desired physiological traits or the generation of desired biological products. Okay, let's break that down a little. Essentially, a GMO is any living organism whose genes have been modified to give us humans something we want. But wait, how does one genetically modify an organism? Let's look at DNA. DNA is short for deoxyribonucleic acid and is the biological molecule that carries all sorts of important information. Think of it as a book that holds instructions for how to produce proteins, regulate metabolism, and reproduce cells. Instead of using words and letters to convey information, this book uses base pairs. There are 4 base pairs which form the rungs of DNA's double helix ladder: adenine, cytosine, guanine, and thymine. These base pairs are complementary, meaning they always pair in the same way: adenine with thymine and guanine with cytosine. The order or sequence of these bases forms specific instructions in the genome. The human genome holds about 3 billion of them. Because of all the ways to mix and match these base pairs, DNA is capable of producing a wide range of useful proteins. Now, let's say we wanted to take a gene from the jellyfish, Aquaria victoria, that codes for a green fluorescent protein, and place it in E. coli to make a glowing green bacteria. Well, to do this, we follow 4 main steps. First, we get a restriction enzyme. A restriction enzyme is a molecule that acts like a pair of scissors, and it's able to cut DNA in specific places. So we choose one that's able to cut out our gene of interest. Depending on your enzyme, this cut leaves sticky ends, which are 4 base pair overhangs. You'll see why these are useful soon. Next, we have to cut our host DNA. Since we're using E. coli, we'll be cutting a bacterial plasmid, which is a circular form of bacterial DNA, using the same enzyme we did before. We now introduce the two to each other, allowing our gene to find itself into the cut area. The sticky ends are particularly useful here, since the 2 pieces are complementary and are therefore attracted to each other. To ensure that the newly inserted DNA stays in place, an enzyme called DNA ligase is added. It acts as a glue, fusing the 2 DNA sequences together. Once we insert this plasmid into the bacteria, it now has the instructions it needs to produce GFP. Who wouldn't want glowing green bacteria? Okay, maybe a lot of people.
But genetic engineering has many more benefits than this. For example, genetically engineered E. coli can be used to help diabetic patients by producing insulin cheaply and quickly. Or genetically modified algae could grow faster to produce more biofuels. Or engineered yeast could be used to produce painkillers. What I'm trying to say is that GMOs have a lot of really cool useful applications. So the next time you go to the store, remember that those GMOs aren't always as bad as they're portrayed to be.